Let us start with a new type of metabolism, and that is nitrogen metabolism, which actually relates to the metabolism of amino acids. You already know what amino acids are. And in this particular lecture, we will also look at one of the most important cycles or metabolic pathway, which is the urea cycle. Now, there are two parts to this lecture. The first part we will talk about transport of nitrogen and urea cycle, and second part relates to the disorders of the urea cycle. For the disorders of the urea cycle, I will not be going through the slides because all the information that you need to know are written on the slides. What I will like you to do is to prepare a table where you need to indicate the disorder, and then you need to identify if it's a autosomal recessive or autosomal dominant disorder, and most of the time it's an autosomal recessive disorder, or if it is an X-linked recessive disorder, what are the predominant symptoms, and in your case you also need to identify the treatment that is availed in order to manage the disorder. And that particular table is very important because questions are asked in different licensing exams and also in the assessments regarding this disorder. That's the reason I made a list of disorders, and I have taken the information from a book known as Mark's Biochemistry of Clinical or Mark's Clinical Biochemistry. So in the first phase, we will go through the lecture slowly. We will try to understand the basic concepts, and also there are some self-check problems for you. So while you are listening to the lecture, you should try to take a stock of things and your learning progress through the lecture. Now, nitrogen metabolism relates to amino acid metabolism, as I mentioned. So the next lecture we will talk about metabolism of amino acids. That's the focus of lecture 20. But most importantly, again, I am not going to go through individual amino acids. In the next lecture, we will look at the clinically relevant aspects of amino acid metabolism, because frankly, the process of amino acid metabolism is quite elaborate, and it's not really possible to go through individual amino acids, and I believe it will be also difficult for you to remember all the different steps of amino acid metabolism. So that's what we are going to do in lecture 19 and lecture 20. So let us look at the roadmap of this current lecture. So what we are going to focus here is on the fact, how does the body transport nitrogen from different organs, this will be from different organs to the site of excretion of nitrogen, where nitrogen is converted to urea, and the site where it happens is the liver. So one particular aspect that you should remember is, most of the time people make a mistake in this, that the site of synthesis of urea is not the kidney but the liver or the hepatocyte. We will talk briefly on the consequence of ammonia toxicity and try to relate it to the TCA cycle. By now you already know what the TCA cycle is. We will talk about different types of carbamoyl phosphate synthetase enzymes. In our lecture here, the synthetase that we will be looking at in detail is carbamoyl phosphate synthetase 1, and carbamoyl phosphate synthetase 2 is actually important in nucleotide metabolism. Most likely they will cover this in the course of molecular biology. Briefly, we will look at the energetics of the urea cycle, and as I told you, I have made a list of urea cycle disorders. I am not going to go through the slides. I'm highlighting this, but please do not ignore that part of the lecture because it's not recorded. It's just that all the information is on the slide and you just need to make a table. And if you make a table, I will like you to show it to me, and maybe one of the tables which one of you make can be shared with the rest of the class so that they can benefit from it. So let us start. Basically, if you look at the structure of amino acid, and by now you should be very familiar with the structure of amino acid. So in the structure of amino acid, this is the alpha carbon. We go back to the first or the second lecture where we talked about this, and then we have the side chain, and then we have the carboxylic acid group, and then we have the amino group. So this particular amino group gives rise to ammonia, as we shall see in this and the next lecture, and that ammonia has to be actually excreted out because ammonia is highly toxic to the physiological system. And that's all, and that is what we will deal about in this lecture, is to transport this ammonia and to excrete this ammonia out of the body. How metabolism of amino acids really happen, we shall look at that in the next lecture. Now, in order to discuss the formation of ammonia, you need to have a basic idea about protein metabolism or breakdown of proteins after you ingest protein. So in the gastrointestinal tract or the GI tract, proteins are digested into amino acids by a group of enzymes which are proteolytic enzymes known as proteases. Now, protein converted to amino acids. The amino acids are actively transported across the intestinal wall into the portal circulation. We will talk about what portal circulation is very briefly, or maybe this has been already talked about by Dr. Nerissa in one of her anatomy lectures, but we will revisit what portal circulation is. And these amino acids are then carried to the liver and also to different tissues, and in these tissues these amino acids are broken down to ammonia. Now, liver is the principal organ responsible for degradation and synthesis of amino acids. So once these amino acids are degraded, they produce ammonia, mostly in the liver, but also to some extent in the other tissues. And in our lecture, we will focus on transporting ammonia from the brain and from the muscle. Once ammonia is produced and transported successfully to the liver converts this ammonia to urea, and this urea is excreted in the urine. That's a simple process of protein metabolism. So proteins converted to amino acids, converted to ammonia, and ammonia is ultimately converted to urea. So here, one aspect that you should note is if there is any kind of hepatic impairment, means the liver has some problem, then ammonia cannot be converted to urea, and that leads to toxicity. That means the person will have some impairment, and most of the time the organ that is affected by ammonia toxicity is the brain. Now, what is the basic mechanism of ammonia toxicity? Now, in our lecture on TCA cycle, we talked about alpha ketoglutarate, which is one of the key metabolites, and it is acted upon by the enzyme alpha ketoglutarate dehydrogenase, and the cycle of the citric acid cycle continues. Ammonia reacts with this particular metabolite. So if ammonia reacts with alpha ketoglutarate, very simply this particular metabolite is used up and the TCA cycle cannot continue. So it makes it a limiting factor for the TCA cycle. And since the TCA cycle does not continue after alpha ketoglutarate, ATP levels go down, and that leads to toxicity. And as I told you, the organ that exhibits detrimental effects of ammonia toxicity is the brain. As I mentioned in the previous slide, liver damage or hepatic impairment, metabolic disorders, this may be alcoholic fatty liver disease or non-alcoholic fatty liver disease where there is severe hepatic impairment, are associated with elevated levels of ammonia, which lead to neurological symptoms such as tremor, slurred speech, blurred vision, and if it is not managed, it ultimately leads to coma and then death. For your information, the normal concentration of ammonia in blood should be between 30 to 60 micromolar. However, people say that it should be maintained below 40 micromolar. So let us say you should have ammonia levels in the blood below 40 micromolar or between 30 to 40 micromolar, but the body can buffer it up to 60 micromolar. If the levels of ammonia increase beyond 60 micromolar, the person will exhibit ammonia toxic effects. So I come back to this old concept again. As I said, amino acids are converted to ammonia. So amino acids can be derived from dietary proteins, that means from your food, and from intracellular protein. For example, certain proteins that are not required, body will break it down and produce amino acids. Also during starvation, body will break down certain proteins and they will be converted to amino acids. These amino acids lead to the production of ammonia, and this is in the physiological system. It is always protonated in the form of ammonium ion. Now this particular ammonia that is produced by the metabolism of amino acids can be reused for the synthesis of amino acids and nucleotides, which are the building block of the nucleic acids that you know, that is RNA and DNA, and also for specific biological amines. And biological amines, you will learn about them in various courses during the program. So this particular ammonium ion can be reused in the synthesis of nitrogen containing biomolecules in the body, very specifically amino acids, nucleotides, and biological amines. Most of it, however, reacts with a metabolite called carbamoyl phosphate. We will talk about this when we come to the urea cycle. And this ammonia and carbamoyl phosphate lead to the formation of citrulline, and citrulline then through the urea cycle leads to the formation of urea, and this urea is excreted out in the urine. The site of production of urea, I mention it again, however, is the liver. Don't make this mistake. Sometimes we think about urea, urine, uric acid, and people make this mistake that they identify that the site of production of urea is the kidney. That's not the right answer. The site of production of urea inside our body is the liver or the hepatocytes. Coming back to this slide, ammonia can be reused, but majority of it is excreted out after being converted to urea through the urine. Now, as I told you, degradation of amino acids in most tissues leads to the production of ammonia. Unfortunately, urea is produced in the liver. So the question that we need to answer is, how do we transport ammonia to the liver from the different tissues? And in our course, we have to study 3 modes. So it is transported in the form of glutamate from all tissues, glutamine from the brain, and alanine from the muscle. So coming back again, ammonia is transported, and we will look into the brain, and we have already talked about the glucose alanine cycle. So from the brain to the liver by glutamine, from the muscle to the liver by alanine, and to a minor extent from the tissues to the liver by glutamate. Other than that, we cannot transport ammonia in any other form. So this is a self-check for you. Which of the following amino acids facilitates the transport of ammonia from the brain to the liver? And the answer is glutamine. You may expect 1 or 2 of this kind of question in your end of course assessment. So that's the reason I have put these self-check questions at different points in the lecture. Now let us look at the transport of ammonia from the brain, and that we have to look at it in detail. Now, there are 2 ways glutamate is used up. So going back a bit, so all amino acids inside the brain will be converted to glutamate, and we shall look at the basic architecture of this reaction. Just wait a minute for a few of the slides. So for now, take that most of the amino acids are converted to glutamate. Glutamate, in the presence of ATP, takes ammonia that is produced by the metabolism of different amino acids, and by the enzyme glutamine synthetase is converted to glutamine. And you can see here, this is the structure of glutamate. You don't need to remember it, but you can see here glutamate contains 1 nitrogen, whereas glutamine contains 2 nitrogen molecules, 2 nitrogen atoms. Sorry. The produced glutamine is transported by blood to the liver. In the liver, glutamine is converted back to glutamate, so this nitrogen is released in the form of ammonia, and this ammonia then goes into the urea cycle to produce urea. So this is the major route by which ammonia is transported from the brain to the liver. So amino acids get converted to glutamate. Catabolism of amino acids leads to the formation of ammonia. Brain has very high levels of glutamate, so this glutamate then reacts with this ammonia, leading to the formation of glutamine. Glutamine is carried by blood to the liver, where it is converted to glutamate, and the ammonia that is released goes into the urea cycle, leading to the formation of urea, which is excreted out in the urine. There is a minor route, and the reason I am mentioning this is because sometimes questions are asked and amino acids may be converted to glutamate. So we have glutamate and this glutamate is then converted to a neurotransmitter, which is gamma amino butyrate, or known as GABA. Gamma amino butyric acid. GABA is a neurotransmitter. Probably Dr. Bakroom would have talked about this when he talked about cell signaling. So glutamate is converted to GABA, and that GABA acts as the neurotransmitter. In general, brain has very high concentrations of glutamate, and glutamate can be produced from amino acids. That pool of glutamate will react mostly with ammonia that is produced by the catabolism of amino acids, getting converted to glutamine, transported to the liver, converted back to glutamate. In the process, ammonia is released, goes into the urea cycle to produce urea. A minor route, amino acids get converted to glutamate, glutamate gets converted to GABA, and GABA is a neurotransmitter, so it helps in brain function or neurological function. So that's how the brain is able to maintain homeostasis. In the muscle, we have already talked about this. I think you already know the glucose-alanine cycle. So here, amino acids release ammonia, and this is taken up by glutamate. We will look at this reaction in detail in the next lecture. For now, just look at the pathway. Glutamate, in the presence of pyruvate that is produced from glucose by the process of glycolysis, is converted to alanine and alpha ketoglutarate. This alpha ketoglutarate can go into the TCA cycle to produce ATP. Alanine comes out into the blood from the muscle cells and is carried to the liver, where it is converted back to glutamate, and here, when it is converted back to glutamate, it leads to the production of pyruvate, and for this process you need the presence of alpha ketoglutarate. Pyruvate, by the process of gluconeogenesis, which we have looked, is converted to glucose, and this glucose goes back into the muscle where it generates ATP. And this we have talked about, as I mentioned, in the glucose-alanine cycle when we talked about the process of gluconeogenesis. So very simply, glutamate in the muscle is converted to alanine. Alanine transports the ammonia to the liver in the non-toxic form, and alanine, if you look at it, provides the carbon skeleton for the process of gluconeogenesis, because it is responsible for the production of pyruvate in the presence of alpha ketoglutarate. So this is a self-check question for you. Which of the following amino acids provides carbon skeleton for gluconeogenesis? And the answer to that is alanine. Now we come to this figure. I will request you to take a printout of this figure. This figure actually summarizes the process of transport of nitrogen inside the physiological environment. We start from this particular part of the slide. So you have glutamate, which helps in the trapping of ammonia that is produced by deamination or catabolism of amino acids. In the presence of the enzyme glutamine synthase, glutamate is converted to glutamine. Glutamine is carried by the blood to the intestine. In the intestine, glutamine is converted back to glutamate, and this during the formation of this glutamate, the ammonia is released, and ammonia by portal circulation is carried to the liver. What is the portal circulation? Portal circulation or portal vein is a vein that capillarizes twice, and you can see here this particular circulation is a direct connection between the liver and the intestine. So ammonia that is produced in the intestine is carried to the liver, where it goes into the urea cycle. A minor part of the glutamine goes to the kidney, where glutamine is converted to glutamate. Ammonia that is released is directly excreted through the urine in the form of ammonium ion, or in the urine you have water. That means it will be converted to ammonium hydroxide. Basic reaction: ammonia plus water, ammonium hydroxide. Now we come to the muscle, and I mentioned that how do amino acids give rise to glutamate, and I told you at that time just wait for some time and we will look at it. So specific amino acids, and these amino acids we will talk about in lecture 20, that is the next lecture. In the presence of alpha ketoglutarate, in the presence of a transferase enzyme, aminotransferase, get converted to corresponding alpha keto acid. For now, just remember this basic reaction, and in the process they produce glutamate. Aminotransferase requires vitamin B6 as a cofactor, very important. Glutamate in the presence of pyruvate is converted to alanine, and this particular reaction you have already seen in the glucose-alanine cycle, catalyzed by alanine aminotransferase. Alanine is non-toxic. It comes out into the blood, is carried to the liver. In the liver, alanine in the presence of hepatic alpha ketoglutarate is converted to pyruvate. In the process, we also get glutamate back, and pyruvate goes into gluconeogenesis or the TCA cycle, depending on the needs of energy or glucose inside the physiological environment. Glutamate, by the action of glutamate dehydrogenase, is converted to ammonia, and in the process it releases alpha ketoglutarate, which further helps in the formation of pyruvate, or this alpha ketoglutarate may also go into the TCA cycle depending on the physiological needs of the body. Amino acids in the liver, I mentioned this in the start of the lecture, that liver is the predominant site for amino acid degradation. Amino acids in the presence of alpha ketoglutarate, and there are specific amino acids, and I told you here that we will talk about them in lecture 20. So amino acids in the presence of alpha ketoglutarate, by the catalytic activity of transferase enzyme that requires vitamin B6, are converted to glutamate and corresponding alpha keto acids. Part of this glutamate is converted to oxaloacetate, and part of the glutamate in the presence of oxaloacetate is converted to aspartate, which goes into the urea cycle as well. But major form or majority of the glutamate is converted to ammonia by glutamate dehydrogenase. This ammonia then goes into the urea cycle. So you have now received ammonia from various parts of the body. So this ammonia now goes into the urea cycle, and the aspartate that is produced from glutamate in the presence of oxaloacetate also goes into the urea cycle, leading to the production of urea. And this urea then comes out of the hepatocytes, is carried by a specific circulation, which they will talk about when you go to the renal system. So this urea comes out of the liver and by a specific circulation is carried to the kidney and excreted out in the form of urine, or in urine, sorry. So this is a very important slide. Try to look at this slide and I guess you will be able to grasp the concept how ammonia is transported from different tissues to the liver. What is missing in this particular slide, of course, is the aspect of the brain, and that I have discussed in the previous slide. I have updated the slides in this lecture, and this is another slide I included, which is a simplistic representation. Glutamate converted to glutamine, carried to the liver, converted to glutamate again. Ammonia released goes into the urea cycle. Part of the glutamine, similar to here, the tissues that we looked at, is also taken to the kidney, converted to glutamate, and the ammonia that is produced is directly excreted out in the form of ammonium hydroxide in the urine. The glutamate that you have transported here is converted partly or mostly to ammonia and partly to aspartate, and from the muscle the alanine that is bringing the ammonia is converted to ammonia and to pyruvate. The ammonia that is formed goes into the urea cycle to produce urea, and the urea goes to the kidney and is excreted out. The pyruvate that is produced from alanine, depending on the energy needs of the body, will undergo either gluconeogenesis or the TCA cycle to produce ATP or glucose as is needed. Now we come to the urea cycle. So we have been able to transport nitrogen from different organs to the liver. Very, very important. Site is the liver. Don't make this mistake, because I mention this all the time, but still people make the mistake of choosing kidney as the organ for the site of synthesis of urea. So in the exam, please remember that the hepatocytes are the site for the synthesis of urea. Till now we have looked into metabolic pathways which involve either the cytosol or the mitochondria. Urea cycle is a typical pathway that involves both the mitochondria and the cytosol. There are 5 steps in the urea cycle. The first 2 steps occur in the mitochondria. The rest of the 3 steps happen inside the cytosol or cytoplasm. What is the key aim of the urea cycle is to convert ammonia to urea. Urea is the predominant disposable form of ammonia and accounts for 90% of the NPN in urine. This is the urea molecule. If you look at it, there are 2 nitrogen, and this is very, very important. One nitrogen is derived from ammonia. The second nitrogen is derived from aspartate, which you have produced in the presence of oxaloacetate from glutamate. And the carbon and oxygen that is present in urea is donated by carbon dioxide. Important facts and simple MCQs are often asked from this particular slide. As I mentioned, the urea cycle converts ammonia to urea, which is a non-toxic substance. One of the nitrogen atoms for urea synthesis comes from ammonia and the other is donated by aspartate. The carbon atom of urea comes from carbon dioxide. And therefore, sometimes people will ask, why is the TCA cycle important for the formation of urea? And the answer to it is that the TCA cycle is generating carbon dioxide, which is donating the carbon atom for the formation of urea. Again, I mentioned this, urea formed in the liver is highly water soluble and is carried by the blood to the kidneys, where it is filtered and excreted in the urine. Basic information, and I will again highlight that this slide is very, very important. This is a simplistic overview of the urea cycle, and as I said, I have updated some of the slides because I thought that the slide of the urea cycle that I have put in the lecture looked complicated and it will be difficult for you to understand it, so I put a simple slide and a simple figure. So as I mentioned, the urea cycle consists of 5 reactions catalyzed by 5 enzymes. The first 2 reactions occur in the mitochondria. The first reaction is the conversion of carbon dioxide and ammonia to carbamoyl phosphate. This rate limiting step is catalyzed by carbamoyl phosphate synthetase 1. So, what is the rate limiting step in the urea cycle? The answer is the formation of carbamoyl phosphate from carbon dioxide and ammonia. What is the rate limiting enzyme? The answer is carbamoyl phosphate synthetase 1. Once carbamoyl phosphate is formed, it couples with a particular molecule known as ornithine. So carbamoyl phosphate plus ornithine, and this leads to the formation of citrulline, and citrulline then comes out from the mitochondria, and also formation of citrulline initiates the urea cycle. I mentioned this over and over again. The free ammonia that is utilized in the step is derived from glutamine by the process of deamination by the catalytic activity of glutaminase or glutamate dehydrogenase. Very, very, very important. I mentioned this when we talked about the transport of ammonia from the brain. The rate limiting step, this particular aspect of the slide is very, very important. And the second aspect that you should note here, this process uses 2 ATP. That means in the first step or the rate limiting step of the urea cycle, I require 2 ATP molecules. Once I have converted carbamoyl phosphate to citrulline, I go to the third step, and in the third step, which happens in the cytosol, you require aspartate. This aspartate comes from glutamate and for the formation of this aspartate from glutamate, you require oxaloacetate. Again, the same aspect of the importance of oxaloacetate in urea cycle is highlighted, similar to what we talked about when we talked about the beta oxidation of fatty acids. So oxaloacetate is a key molecule for the urea cycle as well. In the process of this particular reaction, that is the conversion of citrulline to argininosuccinate, in the presence of aspartate, I am taking 1 ATP, but I am breaking down 2 high energy phosphate bonds. So in this particular step, I have used 2 ATP molecules. Very, very important. I'm not going to go through all the steps, but what I want you to note here is in the first step I use 2 ATP. Here, for purpose of calculation, I also use 2 ATP. So the net use of ATP in the urea cycle in order to make urea is 4. 2 plus 2 is 4. Number of ATP molecules, of course, is 2 plus 1, 3. As I told you, I am not going to go through all the steps. For you, they are not also important. Please focus on these particular 2 steps. And I will identify some specific aspects that you need to look at when you study the urea cycle. How is the urea cycle regulated? Firstly, it is regulated by the availability of the substrate. What are the important substrates that you are using? Aspartate, which is derived from glutamate in the presence of oxaloacetate. Ammonia, which you have transported from different parts of the body to the liver. And of course, the carbon dioxide that is coming from the TCA cycle. Apart from that, the rate limiting enzyme, that is carbamoyl phosphate synthetase 1, is allosterically activated by a specific metabolite known as N-acetyl glutamate, which is formed from acetyl coenzyme A and glutamate. So if I have enough glutamate and of course enough acetyl coenzyme A, I will get this particular metabolite, and the process or the rate limiting step will be activated and the urea cycle will proceed forward. As I told you, there are specific things that you need to focus on when you study the urea cycle. I only talked about the first, let us say the first 3 steps, the first 2 steps in the mitochondria and 1 step inside the cytosol. So which of the following enzymes will be allosterically activated by N-acetyl glutamate? The answer to that is the rate limiting enzyme or carbamoyl phosphate synthetase 1. The next aspect, which of the following amino acids is a nitrogen donor for the formation of urea? I mentioned this, the answer is aspartate or aspartic acid. Which of the following metabolites links the TCA cycle with the urea cycle? The answer to that is fumarate. So let us go back to the simple figure. You see here fumarate. So this links the urea cycle with the TCA cycle. Please focus on these key aspects, and that is all what you need to know from the urea cycle. Of course, don't ignore the first 3 steps, the regulation and the rate limiting enzyme. So I believe this gives you an overview of the urea cycle, and if you look at it, let us go back to the simple figure here. This is the step where urea is produced, and this urea is excreted in the urine. But please do not memorize the whole cycle, it is not required. Now in our body there are 2 types of carbamoyl phosphate synthetase 1 carbamoyl phosphate synthetase 1 and carbamoyl phosphate synthetase 2. In our course, we have carbamoyl phosphate synthetase 1 because it is the enzyme that is involved in the urea cycle. Carbamoyl phosphate synthetase 1 is located in the mitochondria. The other carbamoyl phosphate synthetase 2 is located in the cytosol, and this particular enzyme has nothing to do with the urea cycle. The reason I am highlighting this is because in some of the licensing exams there are questions where they write what will happen to the urea cycle if in a cell system carbamoyl phosphate synthetase 2 is added? The answer is nothing is going to happen. So you have to choose the answer which tells that no effect is going to there will be no effect on the urea cycle. So this is the reason I talked about the two enzymes. For the function of carbamoyl phosphate synthetase 1, you require a nitrogen donor, and that nitrogen donor is the ammonia, because ammonia and carbon dioxide is acted upon by carbamoyl phosphate synthetase 1. In case of carbamoyl phosphate synthetase 2, we also require a nitrogen donor, but that is glutamine. Carbamoyl phosphate synthetase 1 participates specifically in the urea cycle. Carbamoyl phosphate synthetase 2 participates in pyrimidine biosynthesis. As I told you, this relates to nucleotide metabolism, so they will talk about it hopefully in molecular biology. Carbamoyl phosphate synthetase 1 is regulated by availability of substrates, that is aspartate, carbon dioxide and ammonia, and also by the metabolite N-acetyl glutamate, which is formed by the reaction of acetyl coenzyme A and glutamate. This particular carbamoyl phosphate synthetase, that is carbamoyl phosphate synthetase 2, is inhibited by a nucleotide CTP. Here it is activated, and here it is regulated by allosteric inhibition. Carbamoyl phosphate synthetase 1 is regulated by allosteric activation. I will keep this slide with, if I were you, I will be keeping this slide with me as a note, because as I told you, carbamoyl phosphate synthetase 2 is a key enzyme in pyrimidine biosynthesis. Therefore, sometimes people ask questions and most of the time students get confused between these two enzymes. Just remember that for our course, we have carbamoyl phosphate synthetase 1 that is present in the mitochondria and is involved in the urea cycle. Let us look at the energetics. Okay, so in the first step, we have used 2 ATP molecules. So here, 2 ATP. Also in reaction 3, we are using 2 high energy phosphate bonds. As I told you, for the purpose of calculation, this will be also 2 ATP. Here we produce fumarate. Fumarate is converted to malate, and in malate to oxaloacetate, and this reaction leads to the formation of one NADH molecule, not NADPH. So one NADH molecule, which gives rise to 3 ATP. So I use 4 ATP, I get 3 ATP. Therefore, there is a net loss of 1 ATP molecule when urea is synthesized. That means urea cycle is a cycle that will require ATP for its operation. This is a question directly from one of the study guides for step 1. So I just wanted to highlight this. Suramine, used for treatment of African trypanosomiasis, which is called African sleeping sickness. This particular disorder is caused by a protozoa called Trypanosoma gambiense, and there is the human beings are the host, and the vector for this is the sandfly or the tsetse fly. So African sleeping sickness is treated by this very, very famous drug called Suramine, and the company that is having the sole patent for this drug is Bayer. It is a drug that was kept secret for a long time, the molecular formula of the drug. However, in 1924, some of the scientists released the structure of suramine. Incidentally, suramine is still used for the treatment of African sleeping sickness. But the drug has severe, and I mentioned this, severe side effects. And one of the side effects is that it inhibits the mitochondrial ornithine transporter. So formation of which of the following metabolites will be inhibited by this drug? And that was the question. Very, very specific question. Generally, you do not get this kind of specific recall questions in step 1, but sometimes people do ask. In this case, we need to go back to our slide, and here is ornithine. So they are telling that this particular transporter is completely impaired by the drug suramine. So what will be, which of the following metabolites will not be found? And the answer to that is citrulline. And citrulline is one of the options. So I believe when one takes the step 1, they will have some idea about this particular drug. So that's the reason they have put this question in. But this question is related to the urea cycle, so I thought of discussing this with you. Suramine is a very famous and old drug. It is used for the treatment of African sleeping sickness. And a few words about this drug. Recently, this drug has also been proposed for the treatment of autism spectrum disorders, and many a times people make this mistake. It has still not been approved for the treatment of autism. As you can see here in this particular paper that came out in 2017, that is 2 years ago, no drug is yet approved to treat core symptoms of autism spectrum disorder. However, in this small trial, they got some positive results. A bigger trial has been planned for 2019, and you can go to clinicaltrials.gov.us, and you can see that they are recruiting patients for that particular trial, where they will like to try suramine to treat autism spectrum disorder. So an old drug with multiple functions or multiple pharmacological benefits. But I will just ask you to refer to Wikipedia. You can see that the drug has severe side effects, so it has to be really managed well. As I told you, there are a couple of slides on disorders of the urea cycle, and I am not going to go through the individual disorders because, as you can see here, all the information is presented to you. And this, I mentioned that I adapted this from the Marx biochemistry, so it is all there. What you need to do is to collate this information in the form of a table. And please do not ignore these slides because these disorders are asked in different assessments. So if you look at it, you can start from here. So you go slowly, read through the slides and summarize it in the form of a table. You can show me the table, and then we can have one table that can be circulated to the whole class. So that brings us to the end of the urea cycle and the transport of nitrogen. In the next lecture, we will talk about the metabolism of amino acids. Sorry. However, I will be focusing only on specific amino acids that are clinically relevant. Thank you for listening to the lecture. go through the slides slowly, taking notes, because this is a new concept, and probably I believe that we do not have time to go through each of these concepts slowly like we did for glucose or carbohydrate metabolism, but we need to make best use of whatever time we have. So I made a more of a concept map type of lecture. So go through the lecture very slowly, listen to it, make notes, and I am happy to check your notes if you want me to go through them, summarize the information, and I told you what kind of questions you may expect. Focus on the drug suramine and also on the different requirements of the urea cycle. If you have any doubts or clarifications, you can approach me in the class. Thank you very much, and thank you for listening.